Thank you, Tarek. Again, my name is Andrew. I'm a graduate student in Paula Hammond's lab. Today, I'll be sharing with you my project, Layer by Layer Nanoparticles for Enhanced Local Drug Delivery to Glioblastoma. Glioblastoma is one of the most devastating cancers and is the most common primary malignant tumor of the brain. In spite of this, the treatment has remained largely the same over the last 20 years, and the standard of care consists of surgical resection followed by a combination of radiotherapy and chemotherapy with the drug temozolomide. This treatment regimen improves survival by only a few short months, and the long-term survival rate is still very low, between 5 and 6%. So while it's a relatively rare disease overall, it has a disproportionately large impact, resulting in over 10,000 deaths every year in the US. The slow advancement of new drugs over the years can in large part be explained by poor drug delivery. The blood-brain barrier excludes the vast majority of low molecular weight drugs and nearly all higher molecular weight drugs from entering the brain tissue from the bloodstream. Those drugs that do have some blood-brain barrier penetration often result in very high systemic toxicities due to the high doses required to achieve effective levels in the brain. An attractive alternate drug delivery route would bypass the blood-brain barrier altogether with a direct local administration. Such local therapies have strong potential to achieve high local drug concentrations without the broad systemic effects. While this may seem impractical for a disease of the brain, there are several local therapies that have advanced to clinical trials for high-grade gliomas. One of the most promising local delivery techniques that has been demonstrated for glioblastoma patients is convection-enhanced delivery. This is a local infusion technique that bypasses the blood-brain barrier and uses an applied pressure gradient to increase the distribution of drugs throughout the tumor. By co-infusing with a gadolinium contrast agent, the approximate location of the infusion and volume of distribution can be tracked over time and evaluated and adjusted in real time. However, a limiting factor in this approach is the rapid clearance of drugs out of the brain, often over a period of 30 minutes to over 24 hours. So in this example, you can see the gadolinium contrast agent causing the hyperintensity shown here is nearly gone 24 hours after the infusion stops. This means that it's difficult to achieve a sustained therapeutic effect. Additionally, the trade-off with less systemic toxicity may be an increase in local toxicity, as now we're delivering high concentrations of drugs directly to brain tissue where there are both healthy and cancer cells present. Nanoparticle drug carriers offer a promising approach to extend the local dwell time of therapeutics in the brain and can be designed to preferentially target cancer cells over healthy cells. Additionally, encapsulation can protect drugs from premature degradation before they can impart their therapeutic effects. However, the extracellular environment presents a unique challenge for the convective transport of nanoparticles in the brain. The extracellular space between the cells through which nanoparticles must transport is extremely narrow and tortuous, as shown in this electron micrograph, with the spaces between cells shown in red. The gaps between cells are approximately 100 nanometers at most in diameter and consists of a network of negatively charged extracellular matrix. So nanoparticles must be able to fit through these gaps to be able to penetrate the tissue. On top of this challenge is the fact that the fluid in the brain is particularly conducive to nanoparticle aggregation. So typically there are two main ways through which nanoparticles maintain their colloidal stability. Electrostatic repulsion, owing to the similar-like electrostatic charges between nanoparticles, and steric or hydration repulsion, which can be aided by the absorption of proteins onto the nanoparticle surface. However, in the brain, the extracellular fluid has a particularly high ionic strength and low protein content, meaning there are more free ions that can shield these electrostatic repulsive charges and fewer proteins that can provide additional steric repulsive charges, all leading to aggregation, which can result in nanoparticles that are too large to then navigate through these pores. So taking this into account, an ideal nanocarrier for convection-enhanced delivery would have a small size, ideally approximately 100 nanometers or less, although too much smaller than that brings up additional challenges with formulation stability and drug loading. Nanoparticles should have high colloidal stability. Particularly, they should have enough steric repulsion on their own that they're able to withstand the lower ionic repulsion that they will experience in the brain. They should have high cancer cell affinity and ideally high cancer cell specificity over healthy cells. This is particularly important in invasive cancers like glioblastoma, where cancer cells migrate throughout the brain and start to interfere with healthy tissues. And then finally, ideally, nanoparticles will have a visualization method so we can evaluate the volume of distribution and in a clinical setting, make adjustments through techniques such as MRI-guided surgery. To begin to satisfy these criteria, I started using the layer-by-layer assembly technique, where we can take a charged core nanoparticle template and absorb polymers of opposite electrostatic charge. This simple assembly method provides enormous versatility in screening a variety of different surface chemistries quickly and easily. So as a first step, I developed a library of nanoparticles with 22 different outer layers to screen for their interactions with glioblastoma cells in vitro. In this library, I tried to select a range of polymers that had different surface chemistries, such as carboxylated or sulfated charge groups, as well as some that have known interactions with receptors on glioblastoma cells, and others that have been less studied. What I found by incubating this library with glioblastoma cells is there's quite a wide range in their cellular associations, with most layer-by-layer particles having 5 to 10 to even 20-fold improvement over the bare liposome. And in particular, one polyamino acid, polyl-glutamate, demonstrated by far the highest cellular association with glioblastoma cells. However, when it comes to the issue of colloidal stability in the physiological environment, most of these nanoparticles do not pass a simple test of whether they remain stable in artificial cerebrospinal fluid, a common mimic for the interstitial fluid in the brain. And in fact, none of the top 10 performing layer-by-layer nanoparticles were able to maintain that small size in ACSF. And this is not necessarily surprising, as most highly hydrated polymers that are very anti-fouling, that have very high colloidal stability, often for the same reasons, have fewer interactions with cells. So the approach that I took to balance these two competing trends of cellular uptake and colloidal stability was to combine two polymers, one PLE, which had the highest overall cell uptake, but has poor colloidal stability, and a block copolymer of PLE with polyethylene glycol. This has excellent colloidal stability, but the PEG shields many of its interactions with cells. And so by taking these two polymers and mixing them in the same outer layer of one nanoparticle, we can hopefully achieve a blend of both properties, achieving both high cellular affinity and high colloidal stability. To see if it's actually possible to layer two different polymers onto the same nanoparticle at once, I labeled the terminal amine groups of PLE and PLE-PEG with different fluorophores and incubated them in different ratios with anionic liposome coated in a cationic polyl-arginine layer. After purifying the excess polymer and measuring the mass of the two polymers on the different formulations, I find a predictable trend in which the ratio of polymers that ends up on the outer surface matches the ratio that we initially add to the particles. So this suggests that neither polymer is outcompeting the other, and they can actually co-adsorb or co-layer onto the same nanoparticle. I then tested the colloidal stability with several different ratios, with the weight percent PLE-PEG in the outer layer on the x-axis here, and found that a minimum of 40% by weight was needed to maintain colloidal stability in ACSF. so having the same size in water as it does in ACSF, 37 overnight, around 100 nanometers. Incubating these different ratios with glioblastoma cells and then measuring their cellular uptake reveals a predicted trend where the addition of any PEG results in some initial decrease in the cellular uptake, and this trend continues to decrease as we increase the amount of PEG. And so for this reason, I selected 40% PLE PEG by weight as the optimal ratio of these two polymers. To determine if these co-layered nanoparticles have preferential association with cancer cells over healthy cells, I expanded my flow cytometry cellular uptake experiments to include 3 different patient-derived glioblastoma cells and 3 primary healthy cells. As expected, both the core liposome and a pegylated liposome had very little interactions with any cells, and there was a significant increase in all of the LBL formulations. As you increase the PEG density, both the overall cancer cell uptake starts to decrease, and the overall cancer cell specificity decreases as well, indicating or illustrating why we want to use as little PEG as possible to maintain this cell targeting properties. To further motivate these cell targeting properties, I performed confocal microscopy on glioblastoma cells, and hopefully not too difficult to see. As you would expect, the liposome core had very little visible association onto the cells, which is the images on the far left. With only PLE in the outer layer, there is significantly higher cellular association. However, much of it is interacting with a cell membrane rather than getting internalized into the cell. For the case with both PLE and PLE PEG on the same nanoparticle, we see a similarly high degree of association, but a much more efficient internalization. So the initial decrease in cellular uptake that we see with flow cytometry may actually be in part or fully offset by an improved internalization efficiency. A similar phenomenon is observed in 3-dimensional cell spheroids, where LBL nanoparticles with an outer layer of PLE alone have a very high accumulation on the outer ring of the spheroid and don't penetrate as deep, whereas the co-layered PLE and PLE PEG LBL nanoparticle has a much more uniform distribution throughout the spheroid, which is what you would like to see in a tumor in vivo. So then to test these particles in vivo, I incorporated a lipid with a chelated gadolinium on the head group and formed liposomes to have MRI contrast. By doing this assembly technique, we can perform layer by layer assembly over that gadolinium liposome, and this provides very clear and concentration dependent MRI contrast as seen in these in vitro images. In vivo, if we perform an infusion into just a healthy mouse brain and then 24 hours take an MRI image, we can see again clear contrast enhancement where the injection was. And if we then harvest the tissue and section it for cryofluorescence, we can see a very similar pattern of distribution owing to a fluorophore that's on a different lipid in the nanoparticles. So this suggests that this gadolinium tracking technique allows us to visualize the distribution of the nanoparticles themselves and not just free gadolinium. Now, because MRI takes a 3D stack of images throughout the mouse brain, we can segment these regions of contrast enhancement and develop volumes of distribution. So here I'm comparing the volume of distribution for both the core liposome and the co-layered PLE PEG LBL nanoparticle. And what's exciting to see is that the volume of distribution, typically presented as a ratio to the volume of infusion, is approximately 3-fold higher in the LBL nanoparticle system. Generally, a ratio below 1 is undesirable, as that means you're concentrating nanoparticles in one spot in the brain. Anything from between 1 and 2 is comparable to other nano formulations that have been developed, but that don't have cellular targeting abilities. Next, I looked at the retention of nanoparticles in the brain, as this is one of the key motivations for a nanocarrier-based convection-enhanced delivery. So as a comparison, I included two additional gadolinium groups. One, just a free chelated gadolinium contrast agent, which was almost entirely gone within 4 hours. A second, gadolinium labeled albumin, which had very clear contrast and a very large distribution at 4 hours, but was almost completely washed out by 24 hours. In contrast, both the liposome and the LBL formulation had contrast that was visible at both 24 hours and 7 days later, and was particularly remained visible in the co-layered PLE-PEG system. Now, this is an ongoing experiment, so I'm looking to see how long these particles will stay in the brain and to get a more accurate estimation of the half-life. But from some previous pilot data, I've seen that the LBL nanoparticles can be observed 21 days later with an overall decrease in the volume of distribution, but still some visible contrast. So this would be very promising for a surgery that you obviously don't want to do many repeat injections for. A single dose administration would be needed. As one possible explanation for this retention is that LBL nanoparticles are taken up by cells, which is what we see in vitro. So this is flow cytometry data on a mouse brain where I performed an injection of nanoparticles into the right hemisphere and then dissociated both hemispheres separately. And then using the nanoparticle fluorescence of the left hemisphere to set a gate for a nanoparticle positive population reveals that about 11.5% cells in that hemisphere were nanoparticle positive. And this percentage roughly agrees with the volume percentage that we see the nanoparticles covering in MRI. So it's yet to be determined whether these nanoparticles are being internalized by cells or sticking to cells. But based on our in vitro data so far, we hypothesize that they are being taken up by cells, which is part of the reason why they remain in the brain for so long. Finally, all of this previous data has been in healthy mice, and ultimately we want to use this to treat a tumor. However, the situation could be quite different in a tumor as the cellular density can be higher or lower with a different fluid pressure in the tumor as well. So in some pilot experiments, I've grown tumors in the same hemisphere of the mouse brain, which can be seen with a T2 weighted MRI, and then performed the nanoparticle injection. And this is obviously a much larger tumor than you would treat with a single injection. But the goal of this experiment was to see, can we achieve a similar volume of distribution in tumor tissue versus healthy tissue? And so far, while there's a bit more variability, as you would expect, that volume of distribution remains approximately the same. And so this image on the right shows both human GBM nuclei that are grown in a mouse, it's a PDX model, as well as some native mouse nuclei coexisting, and the nanoparticles diffusing between the cells shown in green. And I think this illustrates the goals of my project of having nanoparticles navigate through a very dense cellular environment that consists of both healthy and cancerous tissues. So to summarize some of my conclusions from this talk and future work, layer by layer nanoparticles can be designed to achieve high colloidal stability and high cellular uptake in the brain. Gadolinium incorporation allows us to visualize the nanoparticles over time non-invasively. And the co-layered PLE-PEG LBL nanoparticle system demonstrates increased brain distribution and retention following CED. Some of my immediate future work is to evaluate the tumor cell uptake of these nanoparticles with a similar flow cytometry dissociation experiment, but then labeling different cell types to see, are the nanoparticles being preferentially taken up by tumor cells over the other healthy cells? And ultimately, evaluate the therapeutic effect using drug-loaded nanoparticles. With that, I'd like to thank you for listening to this talk and acknowledge Paula for her guidance and mentorship in this project, as well as a number of members of the Hammond Lab, particularly Dr. Nick Lamson and Michael Morand, who spent many hours doing surgeries with me, and Wei in the MRI preclinical imaging core for all of her help with the MRI and analysis, as well as my funding sources for this project. With that, I'd be happy to take any questions, and you can also shoot me an email.
Got a question?
This was a really lovely talk. Very exciting that they're stable in something other than water. I have two sort of questions / thoughts. One is I'm wondering how you are characterizing size and concentration. I'm guessing perhaps you're doing DLS or.
That's right. Yeah.
And this could be a really great candidate for using an instrument that we have in our core called the Spectrodyne instrument that will give you size information, and it gives you nanoparticle counting in particles per mil. And this is done very nicely in conjunction with DLS. So you can start with DLS, which is faster and easier. And then the Spectrodyne can give you information that is a little bit higher resolution in terms of the size distribution.
Okay.
Just might be an interesting addition to the current characterization that you have.
Yeah, definitely. That would be good to characterize the particles I put in vivo, especially. Yeah.
The other thought was that you said you were interested in looking at whether or not they're internalized into the tumor cells.
Mm-hmm.
And it seems like this could be an interesting opportunity to do some interrogation with EM to really see where they are, if they are inside. And since it sounds like there's a lot of fluorescence labeling going on, there could be an opportunity to do some CLEM type experiments where we can use the fluorescence to go to map where we go for higher resolution EM imaging.
Absolutely. Yeah, that would be very exciting.
Come down and say hi.
Sure.
I agree. That was a really nice talk and a very important problem. I just had some quick questions on slide 17 when you were doing the facts between the right and the left hemisphere. Were you taking a section of it, or were you just taking the whole half of the hemisphere of the brain for that when you had 11%? Yeah.
I trimmed off the forebrain and the cerebellum.
Okay. And then on 18, when you're looking at the fluorescence micrograph, so I mean, I was kind of trying to see what the pattern is for how the nanoparticles were moving. I was just wondering, could you see any kind of pattern? And that's 150 micron snapshot of that brain. What can you make any conclusions from that? And then I have one more question. Sorry.
Yeah. I think one conclusion you can draw from this is just how chaotic the tumor environment is, and it's really not a uniform pore network. There's always going to be a path of least resistance the nanoparticles tend to take. So you do see regions where the nanoparticles preferentially travel. That could be one limitation.
And then I thought that was really cool about trying to balance the different problems you were trying to address. And I just didn't know how much different the ionic strength is of the fluid in the brain. Is it quite a bit than, say, other tissues?
It's comparable to serum, but because there are fewer proteins, there's a lot more unbound ions, particularly divalent cations, calcium and magnesium. And those are really the ones that cause a lot of problems with colloidal stability.
Great talk, Andrew. And one of the questions I had was in terms of your PEG length. So I think you use 5K. Would it make a difference on the length of the polymer that you used in terms of the stability that you could achieve? And a related question would be, what is the max residence time that you're aiming for in the brain? Or like, what is the eventual goal? Like, how long of a drug delivery system are you aiming for? Two
good questions. With the PEG length, I I haven't investigated different molecular weights. Presumably a higher molecular weight would require a little bit less to maintain the same amount of colloidal stability, but that also might reduce cellular interactions more just because it's a longer anti-fouling polymer. And the corollary would be true for a shorter peg density. So I don't know that there would be any advantage, any clear advantage to going longer or shorter, but it would be something interesting to look into. For the ideal residence time, that really would depend on the drug, but certain drugs that are stable for 7 days in vivo, if you could have those delivered and maintained for 1 or 2 weeks, I think that would be extremely beneficial compared to exactly, yeah.
So I got a question online from Zohre. And sorry, feel free to unmute if you want to add anything to the questions.
Oh, hi. Sorry, I just didn't know if that's a possibility or not, but definitely I can ask if you would like me to. It's really great talk. I wonder if you have studied the TME in that context, how are you affecting the monocyte macrophages and microglia in particular? Do you see any changes after your injection or not?
We haven't got to the stage of looking at an immune response yet.
And is your model is one of the versions of GL261 or by any chance CT2A or 005 GSC or any of those models? Do you know exactly which model it recapitulates in terms of the phenotype of human patients?
Sorry, are you asking which glioblastoma model I'm using?
Yes, because each one of those models, if this is in a, for example, C57 background, recapitulate a different stage of disease or if different phenotype of disease. Do you know which one is it?
I'm not sure specifically.
This is in what mouse? Is it injected in what kind of mouse?
This is a human stem cell line injected into a nude mouse.
Okay, so that's probably at best the monocyte macrophages can be studied.
Mm-hmm.
You mentioned a lot about the blood-brain barrier. I wonder if you consider that often when a tumor like GBM, there is no longer really a blood-brain barrier, it's a brain tumor barrier. And material actually gets, if they're size of IgG and so on, get to the brain. And I'm not sure how would you, because you're injecting particles into the brain, and if we have edema in the brain in GBM, that is quite detrimental to the patient. After such injection, do you think that you're going to change the environment such that the edema is increased, or by any chance solid stress is increased, something like that? Or you think it's pretty safe?
I think a full toxicity response is still needs to be performed. I think the question of the blood-brain barrier versus the blood-tumor barrier is an interesting one. That's obviously glioblastomas are diagnosed on that basis of a slightly leakier blood-brain barrier, but to date haven't seen much data showing that there's significant nanoparticle accumulation in either direction across a blood tumor barrier.
I think there is the data on that. Actually, if one injects directly, at least how Glidel was, the material, if it was loaded with any form of anti-cancer drug, it will be shown very rapidly in CSF. That's how it was difficult to use it. In terms of moving to the brain, if it's injected through carotid artery, it actually gets to the brain. But the difficulty in a blood-brain barrier is not just because of leakiness, it's because there is no good flow like any other way that you have a vessel that is abnormal, full of holes in it. So the trafficking is also impaired by the blood pressure itself in that part. So that some of it is already studied in patients undergo various forms of therapies. But I was wondering, since your polymer is a bit different, you have this layer by layer assembly. And it's supposed to not be kind of an adherent system, if I understood correctly. Would you it still float within that space, or do you anticipate it's coming out or just staying in? Any thought in that direction?
There is a possibility that eventually, as nanoparticles are cleared from the brain, they can be traveled along the perivascular tract and end up being washed out with the CSF, or the possibility that they would cross back into the blood-brain barrier. However, based on our observations of the long retention time, I don't believe that's a significant factor at play here.
Okay.
You certainly bring up a lot of interesting points to consider.
Well, this is really a great talk, and thank you very much for that. Thanks a lot.
Thank you.
Any more questions? Good. I think that was great. Thank you for the great discussion. Let's move on to the hot topic. Thank you, Andrew.